So today, what I want to talk to you about is a blink of an eye in evolutionary terms. 300,000 years is a kind of moment in time. For most organisms on this planet, it wouldn't even be worth thinking about. It's such a brief dalliance. But we, of course, as a species, are incredibly young. We haven't been here for very long, and at the rate we're going, we might not be here for much longer. So we are a kind of, in ourselves, a very brief moment in history. And so actually 300,000 years for us feels like a long time, even if in planetary terms, it's the blink of an eye. So the question is, with that very brief slice of time, can we learn anything useful about human evolution by looking at that, and what it might mean for our understanding of who we are today?
The Gresham College lecture that you're listening to right now is giving you knowledge and insight from one of the world's leading academic experts. Making it takes a lot of time, but because we want to encourage a love of learning, we think it's well worth it. We never make you pay for lectures, although donations are needed. All we ask in return is this: send a link to this lecture to someone you think would benefit, and if you haven't already, click the follow or subscribe button from wherever you're listening right now. Now, let's get back to the lecture.
And when we think about evolution, there's a kind of straightforward and simple rule, really, which is that evolution is basically about reproduction. Stuff that has an impact on your reproduction, and most importantly, on your offspring's reproduction, is really important for evolution, and stuff that doesn't is completely irrelevant. So if I look around this hall, for example, there are some people here who are going to be massively shaped by evolution because they're at the start of their potential reproductive journey. They may have children, they may not have children, they may have grandchildren, but all of those things are still to play for. If you're like me, pushing the wrong end of your reproductive life, you're becoming significantly less and less important in terms of evolution. And of course, once you've reproduced and you're post-reproductive, anything or nothing that happens to you is invisible to evolution. And that's an important point. I'm laboring that point because we often ask questions about, why do we have this disease? Or why has this problem come up now? And quite often, the answer to that question is because evolution doesn't care. It's irrelevant because it has no direct impact on reproduction. So broadly speaking, evolution boils down to the question of how many offspring do you have, and how many of those offspring go on to have offspring themselves. And so you can compare that. This is a little example from the British royal family, for example. So here is Queen Victoria, surrounded by a couple of generations of her family. She was very evolutionarily successful, right? There are a lot of offspring here. And actually, if you're a kind of budding historian and you look at the royal families of Europe, you can see a sort of genetic legacy of Queen Victoria right across Europe. That, in evolutionary terms, very successful. Not so great over here. This is William and Mary from the Glorious Revolution. No children of their own, and in fact, Mary's sister Anne, who took the throne, had no children either, and of course, this is why the throne changed hands. So evolutionarily speaking, that lineage essentially went extinct. Why am I putting out pictures of kings and queens here? The point about this is that if we're measuring evolution and measuring impacts, the really important thing to measure are these things here, the kids and their children, and so on and so forth. And we need to think about that in terms of features, behaviors, things that change the survival or reproduction of our children. So it all ultimately boils down to babies, like this little cute one here. She's not so cute anymore. She probably won't be very happy about this baby photo of my teenage daughter being on the screen, but never mind. When it gets taken down on YouTube, you'll know she's filed an official complaint. So it's all about the survival and reproduction of babies. And so if we think about evolution, the strongest forces in shaping all evolution, but in particular human evolution, are those that impact on infant survival and ultimately on their reproductive success. So if we look, for example, at this data from the WHO from a few years ago now, this is a pretty grim slide. This is a slide of what means that babies don't survive. Infant mortality under 5, okay, the leading cause. And if you look at that data, first of all, foremost, I think it's important to say this is actually a tragic indictment of where we are today, because many, many, many of these things are preventable and should not be causing infant mortality, but they are because of inequality and poverty and all the other things that go with it. That notwithstanding, in evolutionary terms, if we look at these leading causes of death, the majority of them are infectious diseases. Okay? So at a first approximation, your biggest risk, if you're a newborn human infant on planet Earth, is something that will infect you early on. And if it infects you and it kills you, that's it. You're removed from the gene pool, your evolution contribution has finished. So we might expect then that if we're looking for evidence of what has shaped human evolution, that these infectious forces will be the place where we are most likely to find that evidence. And to a first approximation then, infection is a big evolutionary problem, and things that overcome infection are big evolutionary successes. So can we learn about those processes over the last 300,000 years by looking at human evolution? And to quickly recap, for those of you who didn't see the previous lecture, and with the caveat that these dates change all the time, this is an incredibly fast-moving field. People discover new fossils, new DNA all the time. So with the caveat, this is probably wrong already and definitely will be by the time I finish this lecture. This, roughly speaking, is how we think of the evolution of our own species, Homo sapiens. So you roll the clock back, say, 600,000 years. This hominin species, Homo heidelbergensis, is roaming Africa. It spreads out from Africa to Europe, where it evolves into the species we know of as Neanderthals. It migrates across to Asia and becomes this enigmatic species about which we know very little other than their DNA sequence and a few tiny fossils called the Denisovans. And then subsequently, about 300,000 years ago, that species that has remained in Africa has evolved again into what we would call anatomically modern humans, migrate out of Europe again, and meet all these other species around the world, and go on to populate the entire planet as we know it today. And this graph, which I showed last time, lifted from the Natural History Museum, shows us up here, Homo sapiens at the top corner, and the fact that we now know that up until about 50,000 years ago, we shared at least parts of the planet with these other species, certainly the Denisovans and the Neanderthals, and who knows, possibly other species too. So the key and important fact about this is that we have a species, anatomically modern humans, that evolved in Africa, that migrated out, they came into Europe, and we have this delightful phrase that geneticists like to use, admixture, which basically means mating with other people. And so admixture occurred, mating occurred between these ancient but modern anatomically Homo sapiens and Neanderthals in Europe. And then again, with this group called the Denisovans over in Asia. And so modern humans are this blend of these hybrid existences. And you can find out much more about that from various sources online if you want. Why am I telling you about this? I'm telling you about this because this gives us a powerful tool to understand much more about how evolution has shaped modern humans. Because we can look at these hybridization events, so these ancient matings between Homo sapiens and other species, and we can look at the exchange of genes, and we can ask which of those genes have had an evolutionary consequence for us as a species, and what does it tell us about the last 50,000 years. So let me explain what I mean by that. So here we have this ancient mating event, and this is a PG lecture, so I'm going to show skeletons. So here we have Homo sapiens skeleton. On the other side, we have a Neanderthal skeleton, very similar, bit stockier, more chunky, various anatomical differences that mean you can distinguish it, but broadly speaking, similar. When any two organisms mate, and hopefully you might remember this from your school biology lessons, each of us carries 2 copies of each of our chromosomes, one inherited from our mum and one from our dad. When you produce sperm and eggs, those separate, each sperm or egg getting one or the other. And then when you mate with somebody else, one of theirs comes together with one of yours, and you get a new combination. Hopefully that's not news to anybody listening to this. When this ancient Neanderthal and Homo sapiens mated, exactly the same process happened. So one Homo sapiens chromosome was in a sperm or an egg. They mated with this Neanderthal. One of their chromosomes came together. And you end up with this 2, one from each parent, just like anybody else. The offspring of any mating, that includes all of us in the room and all those listening, so you have 2 chromosomes. When you produce your own sperm and eggs, those 2 chromosomes that you've got, one from mum and one from dad, do a process called crossing over. So before you produce sperm or eggs, they come together and they exchange bits of DNA between each other. And so what you end up with when you produce, for example, an egg cell is not a cell that has just your mum's chromosome or just your dad's, but they have a chromosome that has, for example, been inherited from your dad, but with bits of your mum's or vice versa. And this is one of the processes by which we get variation. And this is why we don't all look like an exact clone of our grandmother or our grandfather, but even though you've got part of their chromosome, because there's been a genetic reshuffling. That process happened also 50,000 years ago when Homo sapiens and Neanderthals mated. And so what you end up with in the first and second generation of this cross are chromosomes that are a blend of the Homo sapiens DNA and the Neanderthal DNA. So in this color scheme, they look like this. They're not regularly striped, obviously, but they have some genes from the Homo sapiens parent and some from the Neanderthal parent. And the important point is that to a first approximation, that process of shuffling is random. So what you should expect is that these offspring have a random inheritance of genes that were either sapiens or Neanderthal. Now, that offspring is going to go away, and they're going to have their own offspring, and so on and so forth. And what you should expect, if all of those genes are essentially completely neutral, they have no impact, is that over time, the distribution of Neanderthal genes and Homo sapiens genes will remain random across your chromosomes. And that means that if I was going to look today at all of you sitting here listening to this and to sequence your DNA, which is of course possible, what I should see is a totally random distribution of Neanderthal genes across your genome. But that's not what we see, actually. So what we generally see is something that looks a bit more like this. Broadly speaking, most of your genome is a random mixture of Neanderthal and Homo sapiens. So if you're European, for example, like me, then you've got about 2% Neanderthal DNA, and that is, roughly speaking, distributed pretty evenly across your chromosomes, with the exception of occasional patches where you see either no DNA at all from a Neanderthal, you have only Homo sapiens DNA, or the reverse, you're highly enriched for Neanderthal DNA in that area. Why do we see that? We see that because that is a hallmark of the fact that this is no longer a neutral bit of DNA. It's no longer a neutral gene. What it means, so in this example here, for example, here you have a blue patch, which is only Homo sapiens DNA. So what that means is that the equivalent Neanderthal gene has been selected against. It's been a bad idea over the last 50,000 years to have Neanderthal DNA in this bit of your chromosome, because whatever the gene was you inherited there was not helpful, and it has been selected against. Conversely, if you have a patch like this, we have more Neanderthal DNA than by chance. That means something that Neanderthal great-great-great-great-great-great-grandparent gave you was useful, and it has been retained despite this random process. So we can look in modern human genomes, in all of your genomes, and we can look for patches that look like this. And when we see them, we can say, okay, something here has been important over the last 50,000 years. What is it, and what might it have done? And that's exactly what various groups have done, and this in particular is work of Josh Akey, Serena Tucci and others, where they have looked in modern genomes. And what they've done particularly well in this is they have looked in two distinct populations. Modern Europeans, people living, for example, in London today, and modern Asians living in various parts of Asia. And the point about doing two populations is if there is a gene that has been selected for because it benefits modern humans broadly, it should be in both of those populations. And if there's one that's particularly specific, I don't know, it helps you navigate the tube in London, for example, then it will only be in the European population and not the Asian, or vice versa. So when we do that, that is exactly what you see. So here on this graph at the top, what you see is a chromosome. This is chromosome 7, human chromosome 7. And the little red and blue lines are indications of where there are Neanderthal bits of DNA still in that chromosome. And what you see is that, roughly speaking, most of the chromosome, they're pretty random, except that it's pretty clear, I think, that here is a patch where we don't have any Neanderthal DNA. Why is that? That must be, the assumption is that's because whatever was on that chromosome from Neanderthal was not helpful, was not good, and that's actually disadvantaged people carrying it in the sense that they have not reproduced as well as the rest of us, and they have been lost from the population. And when we look in that region, one of the genes that's in this region turns out to be this rather delightful one called FOXP2. And FOXP2 encodes a protein, which you can see here spinning around, which we have known about for some years because this protein is incredibly important for language. So modern living humans, for example, who have mutations in this protein, often have very severe learning defects around language and communication, speech defects, and are not very successful in being able to communicate. If you look ancestrally, modern humans have evolved this gene much faster than chance. It suggests the gene itself is under very strong selection to do something. And it's different to other primate versions of this gene, for example, and it's different to the Neanderthal version. And so the assumption we have here, which I think is a pretty reasonable hypothesis, is that the Neanderthal version of this gene did not allow you to do what these people are doing here, did not allow you to participate in communication in the same way that modern Homo sapiens did. So for example, if you were a hybrid from this Neanderthal Homo sapiens cross 50,000 years ago, you would probably struggle to communicate with other members of your species. And for those of us who've had a very nervous first date, we know that communication is quite important for reproduction. So if you couldn't communicate, you probably didn't reproduce very well. And so those genes have been lost from the population. So this is one example of a Neanderthal gene that we haven't got anymore because it wasn't advantageous. Can we find examples of genes that are the opposite, where that Neanderthal history has benefited us? And of course we can. And so we can look on chromosomes for the opposite pattern. So now we're looking not for those gaps, but for places where the red and the blue genes are very, very dense, more dense than the rest of the chromosome. And you can see here on chromosome 3, one neat example here. But what's clever about this example, I hope you can see this, is that there are lots of red lines there. So these are the ones derived from modern Asian humans. So lots of people in Asia carry this Neanderthal gene, but it's missing from European populations. So the assumption here is that whatever this gene is, whatever this DNA region is here, it has been helpful, it's been kept, but only if you have a lineage in Asia. So there's something here that is, in some way that we don't quite understand, beneficial for survival and reproduction in that part of the world, less beneficial in Europe. And it turns out when you look at this region of DNA, there are several genes in here, but there's a little cluster here called HYAL, hyaluronic acid genes. And these genes encode proteins that are incredibly important for rebuilding cellular structures after DNA damage, and in particular, after DNA damage caused by sunshine, caused by ultraviolet irradiation. So, for example, if you have a poorly functioning version of this gene, you burn incredibly easily. You get very bad sunburn quite rapidly. And so the hypothesis here is that back 50,000 years ago, we picked up this version of a gene from Neanderthal crosses. It confers better protection against ultraviolet radiation. If you're in a grey and misty northern Britain, that is pretty irrelevant. But if you're in a sun-baked Asian desert, for example, it's really quite important to be able to survive sunburn. And so this gene has been retained in the populations that needed it, and not in the pale, kind of rainy populations of Europe, like the one that we're standing in today. So that's an example that goes the other way. And then perhaps the neatest example that I really like of this study is the one here, which applies equally to all modern humans. So now we're looking for a region, this is on chromosome 12, and there is a region here, hopefully you can see it, very, very densely populated with Neanderthal DNA in both populations, so in both European and Asian populations. So here, in this region, is something that we inherited from Neanderthals. It's turned out to be really useful for all humans, or at least all of the ones that we've sequenced, and it's been kept at very high frequency. And this little gene cluster here contains 3 genes, all very closely related, called the OAS genes. And these genes are very, very important because they drive an immune process. And what they do is they detect and respond to viral infections. And it's a very clever system because what this gene does is it keeps under control the very powerful weapons that you use against a virus to avoid them doing damage when you're not infected. When a virus appears, these genes respond and they drive the production of one of these antiviral weapons that we have called RNAs. And this essentially is a degrading enzyme, something that chews up viruses. And as you may know, viruses come broadly speaking, in two flavors. There are some that have a genome that is made of DNA, like our genome, but there are many, many viruses that have a genome made of RNA, a related but different molecule. And so because our genome is made of DNA, but a virus one is made of RNA, that gives you a tool to destroy a virus by targeting that RNA, and that's what this tool does. So when you detect a viral infection, these OAS genes turn on this weapon, this RNase weapon. The weapon is produced, and it comes and it degrades the virus you're infected with. So this seems like quite a good thing to have, and so the hypothesis is maybe the Neanderthal version of these genes is a bit better at dealing with viruses, and so that's why we have kept it so well, and that is exactly true. In fact, it turns out you can do these experiments in the lab. If you have the Neanderthal version of these genes rather than the Homo sapiens version, you are much better at controlling a whole variety of viruses, actually, particularly things like hepatitis C, tick-borne encephalitis, and West Nile virus. And one of the interesting things about this is the virus group that you are particularly good at controlling if you have these genes all belong to a group called the flaviviruses. And so, to me at least, that is quite compelling evidence that at some point between that mating with Neanderthals 50,000 years ago and today, we have been through quite a strong selective pressure in which flaviviruses were a really big problem. And so what happened was people who did not have this extra protection succumbed, died, didn't inherit their genes. Those people who carried this extra Neanderthal gene were able to survive those pandemics, if you want to call them that, and give their genes on. And that's why we see those genes today. The last twist in this rather neat tale, I think, is that these Neanderthal genes that we inherited have apparently helped us survive at some point in the last 50,000 years. But even more importantly, they have also helped us survive in the last 5 years. Because you may know that during the COVID-19 pandemic, there was intense interest in what made individual differences in our response to the virus. And we will all know people who got COVID-19 and carried on as if nothing had happened. They were completely fit and well. And unfortunately, many of us will also know people who were very severely ill or perhaps died. And the question became, quite often, why is it that two people who are superficially very similar may respond so differently to that virus? And so many groups looked at this by sequencing DNA in those different people and asking what genes do people who seem to brush off the virus have that are missing in people who get severely ill. And it turns out that one of those genes, which you can see over here, is exactly this, OAS3. So it turns out that if you have this Neanderthal gene, your ancestors survived some pandemic in the last 50,000 years, and that's why you're here today. But the fact that you're physically here today in this hall listening to me may also be because that same gene has helped you survive the pandemic that we've all been through in the last 4 or 5 years. Quite a salient reminder, I think, that evolution is not all about dead stuff, it's also about what happens today, and understanding it is really, really important. So that all feels and is ancient history. We're talking about things that happened 50,000 years ago, and albeit they may have an impact today. But it's still quite difficult to get your head around Neanderthal matings 50,000 years ago. But the question is, and that I'd like to turn to now, is really, can we see anything that's even more recent than that? And I said at the beginning that evolution works on very slow timescales, usually millions of years. So asking whether we can see anything evolutionarily over thousands of years is a bit of a tall order. But nonetheless, with the power in particular of very, very advanced genetics that we have received over recent years, we're now in a position to start to see things that have evolved humanity over not just a blink of an eye, but an absolute nanosecond of kind of evolutionary time, just in the last few thousand years. And one of the points that I think is particularly notable is that we think of evolutionary events as being kind of standalone, right? We think of, I don't know, for example, humans migrated out of Africa, that was lovely, got to the rest of the world, end of season. But actually, of course, that's not how it works. When something changes, it has a knock-on impact. So, if we start to evolve tool use, for example, that had a direct impact on humans, because we could benefit from that tool use, but it also had a direct impact on other species, for example, trees that were now cut down that would not previously have been able to be cut down. So there are follow-on impacts. In other words, evolution can spawn more evolution. If you think about the evolution of flight in birds, before birds could fly, there was no need for trees to have fruit that could be kind of spread in the canopy. As soon as they had flying birds that could spread it around, it made sense to evolve fruit that might be attractive to those birds high up in the canopy, and so on and so forth. So humans have evolved too. So the question is, have we evolved in a way that has then shaped our own evolution? And the answer to that is definitively yes. And the bit of human evolution I want to talk about is this one, is the transition from, if you like, hunter-gatherer, nomadic, individual humans to the kind of humans we recognize today, people who are urbanized, people who live with agriculture, people who do all these kind of things that society today depends on. And we know actually quite a bit about how this happened. So what we know is that around about 10 or 11,000 years ago, up until that point, most humans had been hunter-gatherers, nomadic, spreading over large areas, no particular pattern. Around 10 or 11,000 years ago, we start to see evidence of people returning to the same place again and again. Maybe that's because they realized that was a good place to be, it was a safe place to be, it had good food, whatever, but they came back to the same place. What we think probably happened was that in their hunting and gathering, they would, of course, have brought food home. They would have spilt food, particularly nuts and seeds and grains, and some of those would have germinated. And then, of course, if you're a savvy hunter-gatherer 11,000 years ago, you realize that actually, if your corn is growing here, you don't have to go out to hunt and gather it and run the risk of being eaten. You can stay here. And so bit by bit, what we now recognize as agriculture evolved. So instead of gathering stuff from the wild, you could sow stuff and you could keep it in one place. Subsequently, people then realized you could do similar things with animals. We started to keep livestock, so on and so forth. And that agricultural revolution has been incredibly important for human society, but also incredibly important for human evolution. And we can start to see, using modern genetic tools, just how important that has been. So the evolution of agriculture has been most intensely studied in an area called the Fertile Crescent, unfortunately, what is now a very troubled part of the world, so ranging from Egypt through to Iraq in a long swathe here. It's important to note that we actually know that agriculture was being evolved in parallel in other places, Asia, for example, the domestication of rice, South America. But actually, because most of the work and science, if you like, has been in this area, this is the area we know the most about in terms of the evolution of agriculture. And what we know is that about 10,000 years ago, people started to domesticate certain key crops. And we can see that in those crops themselves, because you can see hallmarks of very rapid evolution as people started to select for crops that were, for example, bigger or more edible or more durable around 10,000 years ago. So at about 10,000 years ago, in this area, certainly of the Fertile Crescent and elsewhere in the world, humans were starting to develop agriculture. And that had a really important impact on lots of things about being human. And the first one, and one that's often overlooked, is that it allowed us to stay put. Okay? Up until then, for the rest of human history, we'd had to migrate around, because if you need to hunt and gather, you need to be where the stuff to be gathered is. It's no good staying put and hoping that your kind of banana's going to come to you. You've got to go and find the banana. When you start to do agriculture, you can stay in one place, and you can start to put down roots. In other words, you can start to do what we now recognize as urbanization, and build villages, and then towns, and then ultimately cities. And this, for example, is one of the earliest examples of a town in what is now Turkey. So agriculture, the development of agriculture, had multiple impacts. The first one, obviously, is it changed what we ate. Probably not into this rather delicious-looking meal here, but it nonetheless changed quite radically what we ate. And a common misconception is that it must have made things better. Actually, all the evidence suggests it made things worse. If you look at the early nutritional profile of people at this cusp of agriculture, their nutritional profile is actually worse than the hunter-gatherers that preceded them. And the reason for that is that the diversity of their diet collapsed. They went from eating loads and loads of different species to eating a very, very small subset. So they had a much more reliable food source, because they were growing it themselves, but a much more diminished one in terms of diversity. And that had quite an important impact in terms of nutrients, as we'll see in a second. The second impact is that it allowed humans to start to achieve densities, population sizes that we'd never seen previously. Prior to this point in human history, we were very small groups of nomadic hunter-gatherers that traveled around, and you couldn't get very big because you couldn't stay in one place, you couldn't sustain a big population. Now you're staying in one place, you're growing your own food, you can expand agriculture, you can get to high densities. And high densities are very important for society. They're also quite important for some bad things, most notably infectious diseases. And thirdly, we started to live much more closely with other species, not quite like this, but with other species. By domesticating livestock, we started to share our homes, literally share our homes, with all sorts of species: goats, dogs, sheep, cows, so on and so forth. And a whole bunch of species that we didn't intend, but they came anyway: rats, mice, etc., etc. And that mixing pot has had a really profound impact on the subsequent evolution of humans over the last 10,000 years. And let me show you a few examples of that. So here's the first one. This obviously is wheat. This is a crop that we eat. I said just now that one of the things that happened when we moved to agriculture is we diminished the diversity of our diet. We had plenty more calories, that's a good start, but we missed out on things that were nutrients that were not necessarily present in all our plants. So for example, if you eat 50 different species, you're going to pick up nutrients from lots of different species. If you eat 3 species, you may well be missing something that is only produced in that 4 species that you're no longer eating. And that is true. So we know, for example, there is an amino acid called ergothioneine. And ergothioneine, it's actually still a bit unclear why we need it, but we do need it. We don't make that ourselves. And in fact, many species do not make ergothioneine, including most of the major crop species. So we have to get it from things like fungi, for example. So when we switch, and that was not a problem when you were out hunter-gathering and finding your own mushrooms and everything else, when you switched to eating largely grain crops, you suddenly missed out on this ergothioneine. And that impact is now clear in modern human genomes, because the gene you need to harvest ergothioneine from your food encodes a protein, OCTN1, like this, which picks up ergothioneine in your diet and puts it into your bloodstream. And this gene, we can see in modern human populations, comes in different flavors. Some people, present in the audience probably, don't produce very much of this, but many of us have a version that is much more active. In fact, it's 50% more effective at getting ergothioneine out of your food than the other type of the gene. And if you look at where that highly effective gene is found, what you see is a rather wonderful map like this. Those of us in Western Europe are very, very likely to have this, what's called a high affinity gene. People who have a long-standing African ancestry, much less so. And the explanation for this is that that's because the evolutionary history in Africa was hunter-gatherer for much, much longer. Those of us in Europe have been through this agricultural bottleneck. We are the survivors of people who farmed in the Middle East, essentially. And so in order to survive farming in the Middle East, you needed this version of the gene to help you get nutrients out of your food. Otherwise, you died. And so those of us who have survived that process have that high affinity gene because it's able to scavenge ergothioneine from your food. A very nice example, I think, of where agriculture has shaped modern genomes in a way that we had not previously predicted. And in fact, we can go one step further than this, because we can say, so this is testing an individual gene, but we can turn it around. We can say, what is it in modern humans that apparently has evolved very, very fast? And a very neat way to do that was done by David Reich's group, who looks at relatively recently dead human beings. And when I say relatively recently, I mean the last 6,000 years, more or less. And they sequenced DNA from corpses from about 6,500 years ago to about 300 years ago. So this is quite a big swathe of human evolution. And by doing that, you can look at genes that have essentially not changed over that period of time versus those that are apparently changing very, very fast. And the theory is that a gene that is important for something that is changing will itself change. So if you think about, for example, dealing with an infection, your genes are changing fast as the infectious profile changes, whereas the way that you pattern an embryo has not changed over the last 6,000 years. So those genes are not evolving very fast. So what you see in front of you is a graph of the whole human genome, from chromosome 1 right down here to chromosome 22. Every gene, each 20,000-ish genes of the human genome has been looked at, and the question is, how fast are those genes changing? What you see is the vast majority of genes are not changing very much at all, and that makes sense, right? Because most of what it means to be human has not changed over the last 6,000 years. But you have these occasional genes, the tall ones here, that are evolving very, very fast indeed. These are genes that do something that has changed a lot over the last 6,500 years. And one of the very nice things about this analysis is that one of these genes is that one I just talked about, the ergothioneine transporter. So you can see that gene here, it has a terrible name, but nonetheless, you can see that gene is one of these that is significantly evolving faster than chance. But there are some other neat ones here. And this one, it clearly, is the one that is evolving the fastest. And this one is the one that affects all of us today. So if you're sitting here listening to this, scoffing your late-night cereal with a bowl of milk, then you can thank this gene for that. If you're sitting here listening to this saying, I wish I could have that bowl of milk, but it would make me sick, you can blame this gene for that, because this is the lactase persistence gene. And this is a very nice example of evolution shaping evolution. Because, as you will know, those of you in particular who've got domestic pets, cats, dogs, etc., the basic rule for all mammals is that baby mammals drink milk and big mammals don't, right? If you have a puppy drinks milk. When you wean the puppy and have an adult dog, if you give your adult dog a bowl of milk, it will vomit it all back up again, okay? And that is true for most mammals. It is also historically true for humans. Historically, evolutionarily, humans could also not consume milk as adults. We would drink milk as babies, we would wean, and then we would become what's called lactose intolerant, because you would miss the enzyme to digest it. That was not a problem until we started dairying. We started keeping cattle and sheep and other things for their milk. And then it became quite useful to be able to digest that milk even as an adult. There is a mutation that is presumably quite rare in nature that keeps that gene on into adulthood. And for the last million years or so, that's been totally irrelevant. But suddenly, when we started to have a ready source of milk as adults, having that version of the gene was very beneficial. And the time it was particularly beneficial was when times got really tough. So, for example, when in winter, when there's no food around, you were essentially on the brink of starvation. If you had a cow and you could drink that milk and not vomit it back up again, you had a significant chance of survival. If you drank it and threw it back up again and got no calories from it, that was not helpful. And so what we see here is the very, very strong selective pressure to have this version of the gene that allows us to metabolize milk. And for those of you who are listening to this thinking, yeah, but I'm lactose intolerant, I'm sorry, you're one of these people down here who have got the older version of the gene that is not conferring that ability to drink milk as an adult. So this is an example where we have done something, we have evolved dairying, and it has shaped our own genetic evolution quite dramatically. And the other example you can see here is a whole cluster of genes down here, which all do different functions, but they are all to do with the immune system. And I said right at the beginning of this lecture that one of the strongest forces in immunity is infectious disease, because it kills people, and in particular, it kills young people before they have reproduced. And so we should expect genes that confer resistance to disease to be evolving fast, and they are. So there are genes here that are evolving very, very fast. And one of the reasons they have evolved particularly fast over the last few thousand years is because we ourselves have dramatically increased the risk of infection. And we've done that by living very close to each other. We're all here in a fairly crowded hall, for starters, able to spread diseases quite well, but also because we have brought into our lives things that spread diseases to us, either deliberately or inadvertently, like this rather cute, fluffy thing here, for example. Rats, mice, the fleas they bring, the parasites they bring are often damaging to humans, and so that onset of urbanization and agriculture exposed us to new diseases. And in particular, one of the diseases, of course, that these animals brought with them that has been enormously influential in human evolution is this one here. This is the bacteria that causes the bubonic plague. Okay? And this, as you will probably know, the bacteria is carried by fleas. The fleas live on rodents, and so getting exposed to those fleas on either your pets or more likely your rodents that live in your house is the route by which you pick up bubonic plague. It is very likely that bubonic plague has been around for a long time, but it was a very minor evolutionary pressure when we were hunter-gatherers because A, you didn't often come into contact with these things carrying it, and B, even if you did, you were only living in a band of a few people, even if it wiped out your tribe, it was not a major impact in terms of evolution. When you start to live in a town or a village or a city of thousands of people, now you have the opportunity to have proper pandemic plague and a very significant evolutionary impact, because as we know only too well over the last few years, disease spreads fast in crowded conditions. And we know from very good historical data that the magnitude of the impact of these bubonic plagues has been quite astronomical. So, for example, this is a picture of the plague of Ashdod, which we don't actually know if that happened, but nonetheless. But we do know from good contemporary historical data, for example, that the plague of Justinian towards the end of the Roman Empire swept around the coastline of the Mediterranean, and multiple historians from that time tell us that it killed about 1 in 4 people. And bearing in mind, this is the Roman Empire. This is the sort of center of the global universe at that point. So 25% of the people in the Mediterranean was a big chunk of the world's population who died within just a couple of years. And then much closer to home, we're here in London, those of us in the hall at least, and we will all be very familiar with the impact of the Black Death in Europe. The same disease, different name, same disease, same bacteria. And in particular, the Black Death, which peaked around 1350, at least in this part of the world, is estimated to have wiped out between 30 and 60% of the population of Europe. So this is an enormous mortality, right? A really, really strong selective force. And so it stands to reason that the slightest advantage would actually be a really, really big advantage in evolutionary terms. Even if your chance of survival was 1% better than the guy next door, over that kind of level of scale, that's going to be visible in our evolution. And it turns out that is indeed true. And so there is an absolutely fantastic study done by Luis Barrero, which I really love, which uses an amazing historical resource just down the road from here. So down at East Smithfield, just by the sort of Tower of London here, in 1348, Edward III, who was king at the time, was hiding well away from the plague, very sensible, but watching London be decimated by plague. He was a very astute bit of biology, and Edward III thought, this is not very good to have dead plague victims everywhere. What I'll do is I'll buy a big lump of land, I will dig a big hole, and I will tell my soldiers to throw anyone who dies of the plague into that pit, which is at East Smithfield. And then by about 1350, the plague is over in London. They cover over those dead bodies. And, very astute bit of government there. He thought, well, let's not waste this resource. It's a little bit like, HS2 in this country. Let's not waste this resource. Let's think what else we can do. We've got a big pit. I know, we'll put other people in it now when they die on top. So what you have in East Smithfield is a layer of people who died of the plague, and then above it, a layer of people who survived the plague and died of something else a couple of years later. And that is an amazing genetic tool, because you can look at the DNA of those different layers and say, what are the genes that made you susceptible to the plague and you're in the bottom layer, or helped you resist the plague and you're in the top layer? And when you do that, you find them. It's fantastic. So here we have a plot, just like the one I showed you, of all the genes, and the ones up the top are the genes that are significantly overrepresented in the survivors. So if you had that version of the gene, you were much more likely to make it through the plague. You might have got run over by a horse the following year and died, but you didn't get the plague and die. So they were advantageous in helping you to survive. For example, CTLA4 or the ERAP genes there. And unsurprisingly, these genes are all to do with immunity. So, for example, the ERAP genes are used to reveal to the immune system the presence of a pathogen. Having those helped, presumably, these medieval Londoners to say, oh my goodness, immune system, there's a bubonic plague happening here, you need to deal with it fast and help me survive. So that version of the gene helped them survive against the plague all those years ago, and we can see that evolution today. But the last twist to this tale, and perhaps a slightly miserable point to end on, is that this gene, having these genes, is why we're all here today, especially those of you who have long-distant London ancestors. The reason you're alive today is because your great-grandparents had this version of the gene and survived the plague. Unfortunately, you've now got this version of the gene, and that's probably also the reason, if you're sitting there rubbing your knuckles, that you have rheumatoid arthritis. Because it turns out that this gene is really good at triggering the immune system against plague. Unfortunately, it's also a bit good at triggering it when you don't need to, at inducing autoimmune diseases like lupus and rheumatoid arthritis. And so what you end up with is a gene that was massively advantageous a long time ago, now, unfortunately, having a bit of a negative consequence. And of course, the really bad news is, as I said at the beginning, evolution works on reproduction. So it's really important to be resistant to plague and have your babies grow up. By the time you get rheumatoid arthritis, most of us have done our reproduction. It doesn't impact at all on our ability to reproduce, and so evolution doesn't see this, and so you're going to be stuck with this gene probably forever now, because there's no evolutionary pressure to remove it again for the population. Bad luck, but that's evolution for you. Thank you very much. I'm very happy to take questions. Oh, okay, so for those listening online, you can hear that. How has modern medicine changed evolution? Well, I'm going to be really mean and say stick around, because the next lecture, we're going to talk a bit about that, but I'll give you a quick, spoiler alert. So the short answer is it is probably too soon to see anything particularly dramatic, but there are some very nice examples of where we will very soon, I think, see evolutionary impacts. And probably the biggest one is around reproduction, because things like IVF, of course, have just changed this beautiful model I've shown you about. It's all about how successful you are at reproducing, because now you can reproduce if you actually couldn't reproduce previously, or indeed you can choose not to reproduce even though you're biologically capable, which is something that has not happened for millions of years and is undoubtedly going to reshape evolution in some really interesting ways. And that's all I'm going to say. You'll have to tune in for the next lecture to find out some more.
I'm going to go to a question online, then I come to the audience. We have a whole series of questions which I think are kind of points of clarification about genetics, yes? And people are asking questions about how lactose intolerance genes change their expression over the life course, and how they're inherited. Whether if you're lactose intolerant, it means that one of your parents was lactose intolerant. So there's just some people who want some clarification on those points.
Yeah, very good point. So, and I just slightly brush over that. So how does the lactose gene work? So essentially you have a gene, lactase, which helps you digest milk sugars, lactose. If you can't digest the sugar, it makes you sick and you vomit it back up again. So having that enzyme expressed, produced, is critical to be able to digest milk. Genes work, you have the thing that you're making, so that enzyme, and what's called a promoter sequence beside it, which tells the body when to put that protein into production, when to turn it off. So for instance, there are some proteins that are produced at different times of the menstrual cycle if you're female, or when you go into adolescence, or so on and so forth. You don't want the gene for beards being turned on when you're 6 months old. You only want it when you're in puberty. And so those promoters are, if you like, the timer. Lactase, this enzyme, has a timer which says, turn me off forever as soon as you've finished, weaning, as soon as you're weaned, essentially, as soon as you've finished drinking milk from your mother. And that works brilliantly well in all animals, including ourselves. What we have done at some point in evolutionary time is there has been a mutation that has broken that switch, and it stays on forever. No use at all, in fact, possibly slightly disadvantageous, because you're making a protein that's of no relevance. Up until the point when you start being exposed to milk as an adult, then massively, massively important. So that's how it works. And the question about inheritance, so there are two sorts, actually, of lactose intolerance. I should have made that clear. The clean genetic type is that. So if you don't have the enzyme, you can't digest the sugar, you're lactose intolerant. There are secondary issues, for example, allergies to milk, which are nothing to do with that enzyme, and are to do with whether you've been stimulated by a particular antigen, so on and so forth. So I guess the cautionary tale here is don't be listening to this at home and thinking, oh, actually, my milk allergy is all nonsense. I don't have that gene. I'm going to go and drink milk. If you've been told you're milk allergic, don't drink milk, okay, regardless of whether you have this gene or not. So there are different systems. But yes, if you're genetically lactose intolerant, then whether your children are or not depends on the gene of your partner too. Because if your partner is lactose tolerant and they're lucky, they'll get that gene and not yours.
Then on the lactose, my cat loves a nice drop of milk or yogurt or cheese or whatever. And the story I've always heard is that they became domesticated when we took up dairying and they killed the mice and the rats in the dairy. So is there something about cats that enables them to drink milk as adults?
So the question is about cats, and cats that drink milk in particular, and how they got domesticated. And this sort of reminds me of that thing about, you should never work in television with children or pets, right? You should never answer questions about cats. So the domestication of the cat is actually a bit enigmatic. I mean, we know it's quite early on, and as anyone who owns a cat knows, it's a bit unclear whether we domesticated them or they domesticated us. Either way, they are now living with us. So some cats are better at drinking milk than others. So there's some variation there, and I have to say I don't know. Be interested to know if anyone online listening to this can post in the chat whether they know whether that's because of the same sort of mutation or not. Cheese, you mentioned about eating cheese. So when you process milk into something, particularly hard cheeses, actually the lactose is largely destroyed because the fermentation process gets rid of that, which is why people who are lactose intolerant usually can eat cheese but not drink raw milk. So it depends a bit on the amount of lactose too.
Okay, there's a question, again, I'm going to oscillate it. Again, I'm trying to put together some questions here. There are lots of people thinking about what future evolutionary pressures are likely to be. They're talking about changes in diet, changes in obesity. Somebody's mentioned that in Southeast Asia, around Polynesia. People have also said, well, if we have all these pressures, modern day pressures, how does that play out now that there are fewer generations per century? Because we are reproducing older, and so whereas we would have 3 or 4 generations a century ago, now we only have 2.
That's very interesting. I'm take the second part first. So the question is about whether evolution is running at a different clock speed, I guess, if we reproduce more slowly, which we do now. And the short answer is, so yes, generally that's true. So evolution is measured on generational time. So if you have, 20 generations in 1,000 years or 15, that makes a difference to the clock rate. So to a first approximation, evolution is running more slowly. The slight caveat to that is that it's also about the mutation rate in your DNA. And as you get older, if you reproduce at older ages, you carry more mutations, sorry to say, but it's true. So there is a higher chance, I guess, of variability in your offspring. That is not enough to compensate for the change. So it's not that it will stay static. We are still essentially evolving slightly more slowly, but it's not a linear change in that kind of rate. And in terms of the question about what's going to happen in the future, again, it's a bit of a trailer for the next one. I think there are lots of interesting things. My teenage kids would tell me that a key skill for reproducing these days is being able to use TikTok. So maybe we'll see sort of genetic selection for good use of TikTok. I don't know, but there are lots of slightly trivial points. But there are lots of things I think that will change. For example, through most of human history, being able to kind of wield a weapon and kill your opponents was quite a big selective force. Happily, for most of us today, that's not a big selective force. So even weedy people like me can successfully reproduce because I haven't been attacked by someone with a sword, happily, thus far, touch wood.
I was just wondering, could you maybe argue that the most healthy diet is a paleo diet, but then with a bit of milk and carbs? And then the other thing was, in reference to the question of the man behind me about healthcare, could you maybe argue that with the onset of the Industrial Revolution, the infant mortality rate has decreased from like 50% to 1%, less than 1%, and that's caused maybe quite a lot of mutation, and therefore we've maybe become more unhealthy as a population?
So there's a lot in those questions. Yeah. So going in reverse order. Unfortunately, the infant mortality rate globally today is still at about 5 or 6%. That is good in the sense that it has fallen from highs of, if you look historically, it was not unusual to lose half your children before the age of 5, for example. So we've done quite well. I would argue there's still an awfully long way to go. 5% is still a very big number. So there is something about that. In terms of the Industrial Revolution, I think that depends very much. So seeing an evolutionary fingerprint of that would be difficult, I think, because it was not even, right? So there were indeed people who lived in, so for example, smog resistance might have been quite important. Having good lungs, it was important if you were in the middle of smoggy Manchester or London or something. But actually a large part of the population was not exposed to that even during the Industrial Revolution. And so many of us will not have been through that selective pressure. So I think that'd be quite hard to see. In terms of the best, I make it a point of principle never to advise people on the best possible diet. But if I was to say that, so I think it's very tricky to say the paleo diet is sort of attractive because I said hunter-gatherers had this better nutritional profile. That is, of course, at a time when the switch was to subsistence agriculture in a very different way than it is now. Most of us, even people who grow your own, we're actually, fortunately, at very low risk of starving to death because you can still always go down the road to the supermarket if you need to. And so I think the idea that you just kind of roll back the clock and go back to kind of Stone Age paleo diet and everything will be well is a bit simplistic. That notwithstanding, there are certainly things about that diet that would be useful advice for people, like having fresh fruit and nuts and fibre, we know is infinitely sensible, and so that's kind of worth considering. But I would caution against someone thinking, let me live as a Stone Age man and everything will be perfect.
How could mRNA vaccines affect and present in our genome and long-term health, and what negative side effects could there be that we don't commonly speak about?
Oh, that's an interesting question. So mRNA vaccines, which we're all familiar with. So the clever thing about mRNA vaccines is they don't integrate into your genomes. They produce a protein, but they don't get inherited. So if, like me, and hopefully like all of you, you've been vaccinated, quite possibly with an mRNA vaccine, you're not going to pass that on to your kids. It's not inherited in any way. It is a transient, if you like, process. I think there's a sort of second part to the question there, which is theoretically, you could do something that is stably inherited that might be useful. So, for example, you could tweak people's immune system. So one thing you could do, right, is you could go back to those genes that we know have protected against bubonic plague, and you could say, well, maybe we should kind of change those in the genome of people who don't have that version to make them more resistant. There are very plausible reasons that you might want to do that for kind of infectious disease purposes. I think the rheumatoid arthritis example is a good caution against that, because of course you might do something for all the best intentions to change in one way, but you need to be very, very cautious about the unintended consequences about unleashing some secondary effect there. But in terms of the question itself, mRNA vaccines definitely don't affect your genome in terms of any inherited way, and actually I'm a huge fan. I think they're a massively exciting technology that has been hugely beneficial for COVID-19 and hopefully will be for lots of other things in the future. And we'll talk a little bit about that in next year's series. So if you're really keen to come back, see you in 2025 and we'll talk about those.
The map you showed at the beginning with the hybridization events showed one in Australia. Does that mean that the earlier forms of human reached Australia, and if so, which ones?
Oh, Australia is it's like cats, a very contentious topic. Okay, so in terms of interbreeding, what we know definitively is that Homo sapiens interbred with Neanderthals in Europe and with Denisovans in essentially Siberia. Everything else is not known in terms of integration. There is a little bit of evidence about some interbreeding with a mysterious species that we don't know yet. That's still far from determined. The arrival of ancient humans in Australia is a massively contested date, so which is wildly variable. And as far as I know, well, I'm happy to be corrected by those listening online or those in the room, there's no evidence for specific interbreeding in that region. And actually, the entire question of who arrived in Australia when is kind of strongly contested. One of the most interesting things to come out just in the last few weeks, actually, is some whole genome data on native Australian populations, and there is very interesting, very high levels of diversity in those Aboriginal populations, which is very interesting in terms of their kind of dynamics. So, for example, it looks like different groups did not mix that radically early on in the history of Australia, and I think there's a sort of untapped, interesting science question there about the human evolution within Australia that doesn't answer your question about mating with other groups outside of it, though.
I think we have time for two questions. I'm going to take one question here, and then I'll come to you for the last question, because you've had your hand up. Some people are really interested in language. When you showed the Fox P2 and how language conferred potentially a sort of an advantage reproductively. But they've kind of extended it forward in their questions to ask how culture and human culture and art and potentially even religion may have imprinted on our genome and had selection pressures.
Another brilliant question. And again, I feel like some kind of, I keep saying, follow me for the next exciting one, because in a future lecture, we will talk a little bit about that kind of cultural evolution. But let me talk a bit about that now. So language, absolutely. I think language is kind of one of those interesting places where it's not quite as obvious as survival advantages like fighting off infection or growing good and true or whatever. But it's less cultural. There's something more intrinsic, right? If you can't speak, if you can't communicate, it is quite difficult to reproduce. So that's in that hybrid place. I absolutely think that culture itself will also select, and we know anecdotally, right, about certain things. So historically, if you were the wrong religion, your chance of successful reproduction was a lot lower because you might have been burnt at the stake or whatever. But usually, those periods are relatively brief, evolutionarily speaking. I'd be kind of surprised if you could see enough genetics, for example, around religious belief or cultural belief, because they're so short and they're so localized. But I'm always happy to be proven wrong, and who knows in the future we might discover some strong genetic factor for, I don't know, religious conformancy or something.
Okay, final question, I think.
Very interesting lecture. I really appreciate it. I'm not medical qualified, I'm not a physicist, but the impression I get is that modern medical science is causing us to defy evolutionary pressures. Do you think we're in the long term able to keep up with evolution of viruses and bacteria so that we'll always have a solution to whatever befalls us?
So that's a high-pressure question to end on, isn't it? Will we survive forever? Rather than defy evolution, I would say we are certainly changing it. So happily, right? So many of the things that killed babies 300 years ago are not a big worry to us, at least here in Western Europe. And that's a great thing. It clearly changes the evolutionary impact. I think the idea that we've somehow stepped off the evolutionary ladder there is a mistake. There are other pressures now. There are lots of other things that influence whether we reproduce and whether our kids reproduce. For example, cultural things, and they will play a role too. I'm pretty optimistic generally about the opportunity for medicine to deal with big disasters, and we've seen happily, a very strong response to COVID-19. Am I optimistic that we'll be able to do that forever? Not so much. I think historically, most species come to an end at some point. The only question is it going to be in 100 years or 1,000,000 years? I'm voting, well, I guess 100, I'm still going to be gone, so it doesn't really matter. But, let's aim towards 1,000,000 at least. That'd be good.
And on that positive note, I'd just like to thank Professor May again. Your next lecture is when, and that's going to pick up?
That's a good question. It's in about 4 or 5 weeks' time, I think, and we're going to be answering some of the questions that you asked tonight, actually, about the impact of society, human impact, things like antibiotics, disease resistance, and where we might all go from here.
So come back again, find out more, have more of your questions answered. Thank you again.
Thank you.